So I should start by acknowledging that I probably win the award for the worst title of a presentation today. So in case you think that's a mouthful, I guess if I had to add a subtitle, it would be, how does the body know how old it is? So we know there are substantial individual differences in the pace at which we age. So you take two individuals aged 50 years of age, you have a meet-up, and one might look 45 years of age and another one might look 55 years of age. So they've been on this earth the same number of years. So why are they aging differently? Now, people oftentimes say to me, oh, it's good genetics, isn't it? But it actually isn't. In fact, the work that we've done seems to suggest that genes account for a minority of it. So between about 20-30% of difference in lifespan is attributable to the genetics. Okay? If it's not genes, then by definition it has to be something else, and it's going to be your environment. And further evidence that it's not just genetics comes from this identical twin pair. Okay? So these are monozygotic twins. They share the same genetic code. They share the same environment, at least the same early life environment. They grew up together in the same home. And they share age and sex characteristics in common. But you can see when looking at the picture that one is aging more quickly than the other. Okay? Much deeper furrowed lines on the brow, older looking around the eyes, and so on. So if it's not the genes, what is it? We know that as the twins age, they start to experience discordant environments. So, for example, they differ in terms of the lifestyle factors. So they might be physically sedentary, one might smoke, the other one doesn't. They may differ in terms of alcohol consumption and so on. They may differ in terms of things like the socioeconomic position, enforced through things like job loss, for example, housing quality, and maybe the neighbourhood they live in, or the job that they do determines the exposures that they receive. And of course, the environment then will determine the pace at which you're aging. This will influence your biological age. And I want to talk to you about a mechanism through which we think that happens. One of them is actually referred to as the epigenome. So the epigenome is the interface between your genes and your environment, where the two things dance together over time. And I'm not going to labour this too much, but the process is a process that's called DNA methylation. So all those things you do, whether you decide to smoke, whether you decide to be sedentary or not, leaves an imprint in your biology. And one of these processes is called DNA methylation. And I'm not going to labour the point too much, but methylation just determines which genes are being switched on and which genes are being switched off. So on average, as a gene becomes more methylated, you see gene repression, so the gene gets turned off. So I'm not a biologist, and this isn't a biology lesson, but what do we know? Well, we know we've got about 28 million of these methylation sites in the human genome. And we know that only about a third of them actually change with age. And more recently, we've established that only about 1,000 of these sites of the 28 million can be used to derive a measure of your methylation age. Okay? And this is really interesting. So we take a drop of your blood, we measure your methylation age, and then what we do is we ask you your chronological age, and we just do a bit of subtraction. So if you lie on the diagonal here, that means your biological age, as estimated from blood, is the same as the number of candles you blow out in your birthday cake. Okay? If it's positive, you're aging faster than your chronological age, 55, which is like 60. And if it's negative, you're aging more slowly, you're a super ager. So now all of a sudden, we're not thinking of aging in the context of the number of candles you blow out in your birthday cake. We can say, okay, give me a sample of your blood and I'll tell you what you look like biologically. So we've got these clocks, okay, for dating the pace of your biological aging. And you might go, right, that's of interest. Maybe I'd like to know, but do they predict anything? Are they useful, these clocks that we generate? So this is some work that we're doing actually in Tilda at the moment alongside colleagues in the US. What I want to show you here is these are 4 of the different clocks that are out there that people have developed. So Grim Age was named after its progenitor, the Grim Reaper. And the idea is if you are older, according to this clock, so a standard unit increase in Grim Age increases the hazard or the risk of mortality by 60% over a 4-year follow-up period. And this is one at the top DNA methylation biologic risk where we've developed ourselves. And again, it's telling you each standard unit, or you can think of that as years older that you are biologically, it increases your hazard of mortality by 60%. Okay, so why are these clocks useful? Well, making the link to the environmental epidemiology that we're here today, is we almost think of these clocks as a way of dating people's biology in the same way that a climatologist might use tree rings or something like this here to indicate the environment that that tree experienced when it was growing. And just to make this a little bit more concrete, again, this is data this time from the Irish Longitudinal Study on Aging. And what we did is we looked at the correlates of this biological age acceleration. So on the y-axis, we have different risk factors for making you older, right, biologically. And then on the y-axis, it's how much older, or whether you see age acceleration or age deceleration associated with these two things. So if we look at the top here, what this is telling me is that an individual who tells us that they have primary level of education are about 4 years older biologically using this clock compared to someone who has a third level education. So remember, this is just taking estimating their age from their blood. But we talk about inequalities all the time, and this is what we see in the blood. Terrible news if you're a smoker. So if you're a smoker for 30, your current smoker for 30 years or more, you're about 10 years older biologically when we estimate your age in blood. Oh, Sheila. And what I should say is these are all the things just linking to the previous presentation, which is great for you, right? So being physically active, high physical activity associated with epigenetic age deceleration, you're younger on average, right? You can see there if you drink heavily, you're older. But this is really good. Being socially connected, again, is associated with age deceleration. So the more friends you have, the younger you tend to be, at least dated in your, as we estimate it from your blood. And again, things like distress, depression, and so on are associated with age acceleration. In Tilda, we actually capture information about retrospective exposures, things that happened to you in the past. And again, we can date the impact of these types of childhood adversities on the pace at which you're aging. So you can see here, for example, that individuals in Tilda who reported that they grew up experiencing childhood poverty were about 2 years older when we dated them biologically using their blood samples compared to those who said they grew up pretty well off. But you can see also the impact if you lost a parent before 16 years of age, you're about 1 year older biologically as well, as estimated using these clocks. So what I want to say is these new ways of measuring aging are fantastic for us in the era of environmental epidemiology because they give us these tools for quantifying the impact of a whole range of environmental exposures on our bodies, right? And we're talking now at the individual level. Okay? So we've been following individuals for a long period of time. And this one I love, right? So imagine you have an exposure in a data set. Sorry, let me phrase that. You have an exposure that you're interested in, but it isn't measured in your data set. So of course you can't address it. Well, if you've got a sample of the person's blood, what we can now do is derive a surrogate measure of that exposure from their blood, right? So I know that's a little bit complicated, but bear with me. So in Tilda, we're very interested in respiratory functioning. We don't have any direct measure of respiratory functioning in Tilda, but our colleagues in the US got people to blow into the peak flow meter, okay? They also have blood samples. So we said to them, can you give us a surrogate, a proxy for that measure, your peak flow measure, in your study? So what they did is they went off and correlated peak flow performance with the DNA methylation, and then said, here you go, see how well that works in Tilda. So we took it back, and look at how beautifully it works in Tilda. So this is telling me, we didn't directly measure peak flow, we measured it indirectly using your blood, and it's telling me for each standard deviation unit decrease in peak flow, you walk about 6 centimeters per second more slowly. So we didn't measure it directly, but we have a proxy for it. We've got a surrogate for it. And why is this fabulous? Because now all of a sudden we can actually start saying to people, we didn't measure something in the data set, but we may be able to give you something that's an approximation to it if it's been measured well in another study and they also have blood. Okay? And then you start to think about things that we haven't measured until that, but we could do based on their blood. So one of the ones you might be interested in might be lead exposure, might be interested in other heavy metals. Someone mentioned this recently, you should be looking at electric vehicle car brake emissions, okay, the new exposures that we don't know what sort of level, what sort of damage they're doing at a biological level. Now all of a sudden we can start doing this work. So again, as I said, maybe other exposures I'm not aware of. I'm not an environment. I don't do environmental science myself, my background. But what we're trying to enable others is to do this. I just want to finish by talking about TILDA. So I'm the co-principal investigator of TILDA at the moment, alongside Professor Roseanne Kenny. And TILDA's been ongoing since 2009, so it's a longitudinal study. So we've been following these individuals over a long period of time now. So we went to them first in 2009, and we're going to go back to them now in 2024 as the Wave 7 sweep of data collection. That's a good broad period for looking at individual changes in their health status over time. But for the people in the audience, one of the really interesting things that we did was in Wave 3 of the survey, we actually asked people, could you report to us all the household addresses you've ever lived in? Okay, so they wrote this down for us, and alongside colleagues in Maynooth, we went back and geocoded all the addresses, so we know where they've lived over that period of time. Oh, sorry, that was my image. And you can see, so these are all the households geocoded around Ireland. So you do have the level of variability you'd want to look at. If you want to look at the impacts of regional health, and green space, blue space, and so on, you've got variability within the data set to look at that. This is work we've done in collaboration with our colleagues in the ESRI. There's Sean Lyons, Professor Sean Lyons, and Professor Anne Nolan. And this is also work we've done in conjunction with the EPA. But you can see here, this is what we've managed to map already in the TILDA data. So, for example, we've got radon, we've got green space, blue space, water quality, water fluoridation, nitrous oxide, PM2.5, PM2.2, PM10. And we also have noise, and we're mapping to things like light pollution and so on as well. So again, we're already doing some of this work, but the beauty of the TILDA data set is because it's got these really detailed physiological measures, biological measures. We can actually look at the impacts of the environment on the aging process. And I'm amazingly 2 minutes ahead. I've never finished on time, so clearly the clock works. But I just want to finish by saying TILDA can be used to explore these long-run effects of environmental exposures. Let's come back to Hans' idea of the exposome, things you experienced in the past. We can look at the impacts of those on your current level of biological aging in the TILDA data set. TILDA is enriched by linkage to other sources of environmental and climate data. So we're hoping to do things like precipitation, ambient temperature, other types of pollutants you may be exposed to, and then maybe just look at the potential for flooding risk and so on. And this is really nice. Alan referenced the pollution study. Sorry, I think it was actually Laura referenced the study earlier that looked at the impact of the smoky coal ban on respiratory function in Ireland. That actually used TILDA data. And the beauty of that sort of thing is because TILDA is a longitudinal study, you can look at the impact of policy changes on health at the individual level. Because we measure pre and post measures, you can actually start to see, does the policy change make a difference? And you can think about some of the ones we have now, the recent public transport initiatives, the active travel and so on. Followed into the future, we're able to look to see, do they have an impact on the individual's health? So with 1 minute remaining, I'll finish there. Thank you.